Hello everyone and welcome back to AP Bio with AP Playground. Today we're reviewing Unit 2, Cell Structure and Function. There are some key points you need to know in Unit 2, and these include the difference between prokaryotic and eukaryotic cells, the organelles of the eukaryotic cell, cell size and why it matters, the structure of the plasma membrane, and the different methods of transport across the membrane, including passive transport, active transport, and osmosis. Some math skills you'll need to know for this unit are calculating the surface area to volume ratio, as well as the water and solute potential equations, and lastly is compartmentalization. Unit 2 was broken up into 11 subunits covering the different cell structure, all the organelles, and then moving on to the cell membrane, and we'll be reviewing all of that here. Unit 2 also covers 3 big ideas, including evolution, explaining the origin of eukaryotic cells, energetics, mechanisms for transport across the membrane, how they use energy, and the advantages and disadvantages of compartmentalization, and lastly, big idea 4, systems interactions, how living systems are affected by the presence or absence of subcellular components, which all ties into these key points within Unit 2. First up, we've got to review the cell organelles. And what is a cell? First of all, a cell is the basic living unit of life. It's not the smallest possible thing that we can break compounds down into. Those would be atoms, and beyond that, subatomic particles and quarks. But the living unit, the basic supporting structure of life, is a cell. And all cells are going to have a membrane, genetic information in the form of DNA, ribosomes, and an interior fluid known as the cytosol. Now, there are two main kinds of cells, and these are prokaryotic and eukaryotic cells. Prokaryotic are the simple, single cellular organisms. They are going to have a single circular strand of DNA, and they're not going to contain any organelles. So they're not a very complex form of life. They're going to have these basic things that all cells have, but not much beyond that. Whereas eukaryotic cells are generally going to be part of multicellular organisms such as humans and animals. They're going to be very complex. They're going to be much larger. They're going to have lots of DNA. It's going to be linear, and it's going to be located within a membrane enclosed organelle known as the nucleus, and it's going to contain lots of other organelles as well. So within 2.1 and 2.2, we have to know about all of these different organelles. Starting with our genetic storage information, this is going to be our brain of the cell. This is the nucleus. So it stores the genetic information within eukaryotic cells. Remember, prokaryotic cells don't have them. And it's got a couple of parts. First of all, the outside membrane is known as the nuclear envelope right here. The actual genetic information inside is located within the chromatin. That's all this stuff over here. And there's also nuclear pores within the nuclear envelope, which allow for the export of messages from the nucleus to be sent to the cell for, let's say, creating some sort of protein. And it also allows ribosomes to leave the nucleus because they are created within the nucleolus right here in the very center of the nucleus, and then are exported out into the cell for use through the nuclear pores. Now, what is a ribosome? Basically, ribosomes make proteins, and they're not really considered organelles. They're not membrane bound, and they're located both within prokaryotic and eukaryotic cells, but they still produce, they still do a specific function in the cell, and they produce proteins. They can either be free or bound. Free meaning they're just hanging out in the cytosol, or bound meaning they're attached to the endoplasmic reticulum. So what is the endoplasmic reticulum? This is the production center, and it consists of membranes and sacs, and it's located near the nucleus. You can see part of the endoplasmic reticulum next to this nucleus, and here's a better picture. And there are two different kinds. It's going to consist of the rough endoplasmic reticulum, which is going to have all of the ribosomes bound on it. That's why it's considered quote-unquote rough. And the smooth endoplasmic reticulum, which is not going to have any ribosomes bound on it. And the portion, the rough endoplasmic reticulum, is going to be basically the protein production center of the cell, while the smooth endoplasmic reticulum will be used for producing proteins, as well as detoxification of objects within the cell. And again, these are just a few of the organelles within the eukaryotic cell. We took a look at these right here, and on the next slide, we'll cover the rest of the organelles within the cell. Continuing on, we've got the Golgi apparatus, and this is the modifier, this is the packer, this is the shipping machine of the cell, this is the UPS, and all things that are going to be exported out of the cell or just even modified within the cell, such as proteins and other supplies, they're going to be entering through the cis phase of the Golgi apparatus, passing through and then exiting the trans phase. And the Golgi apparatus is one of the key steps in the quote-unquote protein production chain that shows how all of the organelles work together when there's a signal being sent from the nucleus to the rough endoplasmic reticulum that the ribosomes need to produce a protein. Then this protein is going to be sent to the Golgi apparatus for later export out of the cell and through the membrane. Next up, we have the mitochondria, which is the energy-making machine of the cell. You may have heard that it's the powerhouse of the cell. Now, the mitochondria consists of a double membrane. It's got an interior section known as the matrix, and it's got all these membrane folds on its inner membrane known as cristae. And what the mitochondria does is it takes food and it's able to get energy out of it in the form of ATP. And we'll talk about cellular respiration in Unit 3. Next up, we've got chloroplasts, which are energy storing machines. These are going to take the energy from the sunlight and turn it into food. And they also contain a double membrane, just like mitochondria. Their interior fluid is known as the stroma, and they're going to consist of these discs known as thylakoids, which contain the pigment chlorophyll. And chlorophyll is what makes plants green. And these thylakoids are going to be arranged in stacks known as grana, which you can see right here. Next up, we've got lysosomes, which are acid death sacs of digestion. These are going to be used for anything that's old, anything that's broken, anything that's not being used within the cell, so that it can be digested and the parts can be reused for other supplies within the cell. Next up, we've got vacuoles, which are large vesicles used for storage. Now, a vesicle is just a membrane sac, and vacuoles are going to be used for storage most notably within the plant cell, and its large central vacuole, which among other resources is going to store water, which the plant cell needs to keep its shape and also just to have as a resource. Now, what holds the structure of the entire cell is the cytoskeleton, and these are fibers and proteins which provide structural support and motility, so they can also assist with movement. And they consist of microtubules, microfilaments, as well as the cilia and flagella, which are small movement objects outside of the cell that can propel. Lastly, we have the cell wall, which is used for protection and structural support, and it is only located within plant cells. Animal cells only have a membrane, while plant cells have both a membrane and the cell wall. These were the cell organelles. Now it's time to talk about cell size. And this is the surface area to volume ratio, SA to V ratio. If you don't remember your formulas for finding surface area and volume of different shapes, never fear, it's on the it'll be given to you on the test, and it's on the College Board course exam and description. You'll be able to see everything there, but you should still practice it if you're not very familiar with some of the equations, just so you know, if in case you get a question on this topic for any specific shape, you're able to solve things out. And basically, you're dividing the surface area by the volume for this ratio to find the amount of surface area per unit of volume. And smaller objects tend to have a higher surface area to volume ratio. We can see this with the example down here, this cubes. So first of all, we have the small cube 1 by 1 by 1. The volume is going to be 1 centimeter cubed, and the surface area is going to be 6. The volume being length times width times height, so we just multiply 1 by 1 by 1, and then the surface area, we take each face of the cube. In this case, the area of each face is 1, and there are 6 faces, so we multiply that by 6. So the surface area to volume ratio here is a 6:1 ratio. Now, moving on to a 2 by 2 by 2 cube, we see that the surface area is 24 centimeters and the volume is 8. The ratio has now gone down to 3:1, and going even higher with a 3 centimeter side cube, surface area is 54, volume is 27, the ratio has gone down 2:1. So smaller objects are going to have a higher surface area to volume ratio. And for cells to exchange enough resources and energy with the environment to support themselves, you need to have a large surface area, because then you're able to have more reactions happening at the same time, more things can go in and out of the cell at the same time. But large cells need more resources and don't have as good of a surface area to volume ratio, because they're going to have a larger volume, so they need to be supported by more resources, and they're not going to have as good of a ratio for the surface area to volume. So small cells get the best exchange for resources for their size. They have a large ratio and they don't have many, they're not that large, so they don't need as many resources. The smaller the cell, the better the surface area to volume ratio. And you can achieve even more surface area through membrane folds, because this isn't going to change the volume. You're not putting more membrane in the cell, you're just folding it up to create more sides. This is going to increase the surface area as well. So why are cells small? This is to maximize the surface area to volume ratio. Now it's time to go to the cell membrane, the plasma membrane. This consists primarily of phospholipids, which have a phosphate head and two fatty acid tails. And the phosphate head is going to be hydrophilic, it's going to be water loving, and the hydrophobic fatty acid tails are not going to like water. They want to get away from water. So they're going to be arranged in a bilayer, meaning there's a layer and there's going to be two layers of phospholipids within the membranes, and this is going to be with other proteins. And in total, this makes up the plasma membrane, or the fluid mosaic model. Now, why are they arranged in this way, where we've got one phosphate head pointing up and the other one pointing down? Why didn't they arrange themselves with the phosphate heads pointing in the same direction? The reason is because of the hydrophobic and hydrophilic sections. Outside of the membrane, there's going to be water, an aqueous solution, something that's going to have water elements in it. So there's going to be water on this side of the membrane, and there's going to be water on the other side of the membrane within the cell, an aqueous solution, the cytosol. So the phospholipids are going to arrange themselves so that the water-loving section is going to point out towards the areas with water outside of the cell and inside of the cell, while the fatty acid tails, which like to get away from water, are going to be pointing inwards. Now, other things in the membrane include integral and peripheral proteins. These are proteins that either go through the entire cell, they're integral or transmembrane, or just through part of the membrane. These are peripheral proteins. Additionally, there are also going to be glycolipids and glycoproteins, which are lipids attached to carbohydrates, and proteins attached to carbohydrates, which can be used for signaling and cell-to-cell recognition. You'll see them at the top of the membrane facing out of the cell here. And lastly, steroids like cholesterol. And these will be within gaps between phospholipids, and they can increase the fluidity or rigidity of the membrane depending on what need be. Because all of these phospholipids, they're not tied down. They're just arranged in this way, and they're not moving much because the hydrophobic sides are away from water and the hydrophilic sides are towards water. So it's really fluid. Everything is sliding along each other, and cholesterol can help increase how rigid or how smooth this sliding along is. And that's why overall it's called the fluid mosaic model. And this membrane has selective permeability due to its structure. Some stuff can go through and some can't. So now let's take a look at things going through the cell membrane. Here we go, passive and active transport. And we're going to be taking a close look at what things can pass freely through the membrane and what can't. So passive transport is the movement of solutes across the membrane with the concentration gradient, from a high concentration to a low concentration. Basically, if there's going to be a high amount on one side of the membrane of a certain substance, it's going to move across the membrane so that eventually there's going to be an equal amount on both sides of the membrane until dynamic equilibrium is reached. So even though things can be moving across the membrane, overall there's not going to be more on one side or the other. This is moving with the concentration gradient from a place of high concentration to a place of low concentration. So first, for passive transport, we have simple diffusion, which is small nonpolar molecules and small uncharged polar molecules, and these can pass through the membrane by themselves. Firstly, they're small, which means that they can fit into the gaps between phospholipids, because of course phospholipid bilayer is fluid, it's moving along, it's not rigid. And second of all, they're nonpolar, they're uncharged, so they don't have a charge which would interfere with their passing through the membrane because of the differing charges and hydrophobic sections and hydrophilic sections of the phospholipid bilayer. Now, for large polar molecules and ions, membrane proteins are needed, which pass through the whole membrane, these transmembrane or integral proteins. This is through facilitated diffusion. Facilitated meaning helping. So the protein is going to help this substance pass through the membrane either through a channel protein or a carrier protein. So channel protein just allows the substance to pass through. It's like a tunnel, and it's generally used for ions, while carrier proteins take the specific molecule and deposit it in or out of the cell. And all passive transport doesn't require an additional input of cellular energy in the form of ATP because everything is moving with the concentration gradient. Meanwhile, active transport is the movement of solutes across the membrane against the concentration gradient. This goes from a low concentration to a high concentration. So even though there are few amount of solute on this side of the membrane and a lot on the other side of the membrane, things are still going to be pumped to the side with a lot already there. And this is going to require ATP, cellular energy. So over here in this diagram, we can see we're using a membrane protein to help with active transport. There are only 3 molecules on this side of the membrane and 6 already here, but things are still being pumped across in this direction, filling up the space. But because it goes against the concentration gradient, cellular energy is needed. And this can use membrane proteins, but for large amounts of solute or other compounds, there's going to be a need for endocytosis and exocytosis, which just uses the folds of the membrane, and it's another form of active transport, so it's going to use energy. And basically the membrane is going to fold around and grab certain supplies. In phagocytosis, this is going to be generally molecules. In penocytosis, this is going to be more liquids, not so much solid stuff. And receptor-mediated endocytosis is only going to be activated once the signaling molecule is going to be bound to the receptor. And then these things will be taken in, the membrane fold will pinch off, turn into a vesicle, a membrane sac, and the stuff will now be in the cell. And exocytosis, the vesicle can bind to the membrane, and the membrane will open up at this fold, and now all of the stuff can exit the cell. So this has been passive and active transport. Now, another kind of diffusion across the membrane, osmosis. And this is the diffusion of water, and because water is a polar molecule, it happens through aquaporins, which is a special membrane protein specifically for water. And water is always going to travel to be proportional to the amount of solute on either side of the membrane. In an isotonic solution, the amount of solute is going to be equal on both sides of the membrane, so water is not going to travel in a net movement. It's going to flow in and out of the cell, but there's not going to be a specific change of movement because there's the same amount of solute. In a hypertonic solution, there's more solute in the environment, so water is going to flow out of the cell. This could cause the cell to shrink because water is leaving the cell and because it wants to be proportional to the amount of solute within and outside of the cell. Meanwhile, in a hypotonic solution, such as fresh water, there's going to be more solute inside the cell, so water is going to flow in the cell, and this could cause the cell to burst. Now, this occurs differently in animal and plant cells because plant cells are going to have a cell wall. So they can become what's known as turgid. This means they're being filled up, but the cell isn't going to burst because of the cell wall. When it's a hypertonic solution, the cell will become plasmolysed, meaning the membrane inside will shrink, but the cell wall itself will remain sturdy. And in an isotonic solution, plant cells are going to be flaccid because they do like to have some pressure. They do like to be in a more hypotonic environment. Now, animal cells are going to be normal at an isotonic environment, completely shrunk in a hypertonic environment, and lysed or kaboomed at a hypotonic environment. And water is going to move from a high water potential to a low water potential, which means it's going to move from an area where water can be free to bind to a lot of molecules placed without much solute to low water potential where water is already bound to a lot of solute. There's not much freedom moving around. So basically from a place of low solute to high solute, water is always going to follow the solute. Now let's take a look at the equations for calculating water potential and solute potential because they may appear on the AP exam. So water potential, which we just talked about to calculate, is the addition of solute potential and pressure potential. Now, because generally you'll be dealing with open containers, the pressure potential will be 0, but if there is a certain pressure potential in the problem, then they will tell you. Now, how do you calculate the solute potential, or what's known as the osmotic potential? The equation is negative ICRT. And what this means is we're taking the negative times the ionization constant, how many ions are present in the solute, usually only between 1 and 2. You're taking then the molar concentration, which is going to be in moles per liter. That's just a way to measure molecules. There's going to be a pressure constant, which is always 0.0831. And lastly, the temperature in Kelvin, which is just the temperature in Celsius and adding 273. Now, both of these equations will be given to you, but you do need to know how to use them. So here we have an example. We're finding the solute potential of 1 molecule of sucrose at a molarity of 0.5 moles per liter at 30 degrees Celsius. So let's set up our equation. Negative I, the ionization constant, sucrose isn't going to split up into any ions. It's just carbohydrate, so the ionization constant is 1 times our molar concentration of 0.5 times our pressure constant of 0.0831, and then lastly our degrees in Kelvin, our temperature in Kelvin, which is going to be 273 plus the degrees in Celsius, which comes out to be 303 degrees, and that should give us a total solute potential of negative 12.58965, so forth and so forth. You round to how many ever units or digits the problem asks for, and you've got yourself your solute and your water potential. Lastly, we need to talk about cell compartmentalization and the endosymbiotic theory. So, compartmentalization is the splitting of the cell so that different reactions take place in different areas. Benefits of this are A, it's going to separate reactions, which allows for enzyme efficiency. Every enzyme that performs a reaction is going to have a specific pH and temperature and salinity that it works best at. So when we have compartmentalization and splitting of the cell, each enzyme can be in the optimal environment so that it'll work most efficiently, it'll work the fastest. And in eukaryotic cells, compartmentalization occurs through membrane folds, as well as the different membranes of cell organelles. So for instance, when we're looking at the lysosome, because it's an acid death sac, the enzymes, the hydrolytic enzymes within it, are going to operate at a much lower pH than the rest of the cell. And within the lysosome, because it's got a membrane, first of all, this environment can be created, and second of all, the rest of the cell will be safe because there isn't going to be this pH, this acidity, spilling out into the rest of the cell and ruining the rest of the enzymes that work at a higher pH that aren't so acidic. And compartmentalization can also increase surface area for reactions because of the amount of these membrane folds and these membrane organelles. This is why mitochondria have cristae, these inner membrane folds, so that they can increase their surface area, they can have more efficient reactions. And double membranes of the mitochondria and chloroplast assist in this. Additionally, ion gradients can be formed. So besides the membrane folds, double membranes also increase the surface area. And lastly, between these membranes, an ion gradient can be formed as more hydrogen ions move against or across the membrane, which we'll see within mitochondria and cellular respiration. And again, because of the movement of hydrogen ions, there's going to be changes in pH on either side of the membrane, but because of this presence of the membrane, the reaction is going to be separate. So enzymes are going to be efficient, and the rest of the cell is going to be safe and unchanged. So what is the endosymbiotic theory? This is the idea that cell organelles originated from free-living prokaryotic organisms, and it helps explain why mitochondria and chloroplasts have the double membrane, why mitochondria and chloroplasts have all these DNA and ribosomes within them, and it explains basically cell compartmentalization, why they are their own organelles. So the idea is that there was an ancestral prokaryote, so it's just going to be the membrane, it's just going to have its genetic information, and eventually a nucleus started to form and there was an infolding of the plasma membrane. Now this ancestral eukaryote with its nucleus took in some bacteria and formed a relationship with them. One was energy producing and one was photosynthetic, and it was taken up by the early eukaryote, and now there's a beneficial relationship for both. The bacteria gets protection and food, while the larger eukaryote is now able to obtain more energy from the energy producing bacteria, from the energy storing photosynthetic bacteria. And eventually these guys became organelles. And that's our ancestral eukaryote with its nucleus, chloroplasts, and mitochondria all the way over here on the right. So evidence for this theory is the mitochondrial and chloroplastic DNA, as well as the double membranes, because they gained a second one as they were engulfed by the ancestral eukaryote. And this just explains how some of the cell organelles within the cell came to be and how some of this compartmentalization stuff actually started to happen. So this was a pretty big unit covering cells, but this is the end of Unit 2, and we'll see you in Unit 3 to talk about cellular energetics.